Welcome everybody. Good afternoon to everybody and good morning for those that are connecting from the USA or from Brazil or Argentina. So today we have our speaker is Daniele Cornara. He also presented in the previous seminar, but today he will talk about a different subject. So Daniele works at the University of Bari in Italy, and today he will present his last research on host feeding and probing behavior of Philaenus spumarius on olive varieties, differing in the degree of susceptibility to Xylella fastidiosa. So Daniele, thank you again for providing us with your ideas and your research. So the floor is yours.
Thanks, Blanda. I'm gonna share the screen. Yeah. Pick up the other time. Now you should see my full screen, right?
Yes.
Okay, cool. So thanks again, Blanca, for introducing me, and to the organizer for inviting me to this second talk in a row in a month with this very long title, Probing Behavior of Philaenus on Varieties with Different Degree of Susceptibility to Xylella. And I just realized that maybe this very long and boring title is the wrong title for this talk, because of a better title would be what vectors of Xylella like of a plant, what makes a plant a suitable host for Xylella, so a suitable host for Xylella too. So I will start with a slide that I prepared for the last seminar on the finding of ST1 in Bari. And the title of this slide is What Moves Xylella Fastidiosa? Because of the problem, at least my perspective, that most of the people when talking about Xylella fastidiosa, they really focus on the interaction plant-bacterium, overlooking at a very important factor, which is what permits Xylella to moves around and to establish and spread, which are xylem-feeding insects, the vectors of Xylella fastidiosa. So compared to the knowledge that we have on the plant-bacterium interaction, the knowledge on the vector-plant-bacterium interaction lags considerably behind the knowledge bacterium-plant interaction. And this is the problem. This is a problem, for example, because of, let's look at Sandys-Purcell equation presented in a paper in 1981. The probability of Xylella to be transmitted is depend on four main factors, which are the vector infectivity, the transmission efficiency, plus this NT, which is a combined factor, and which is the number of the vector on a host plant per the time that these insects spend on the host plant. And this factor combined is called vector activity. And the vector activity is especially important for insects with a relatively limited transmission efficiency of Xylella fastidiosa. If we look, for example, at the transmission efficiency of Xylella exhibited by the blue-green sharpshooter in California from grape to grape, it's more than 90%. If we look at the transmission efficiency displayed by our vector, our main vector, Philaenus spumarius, from grape to grape, it's around 20%, from 15 to 20% in 24 hours. If we look at a review by Bodine and colleagues in Turin, the transmission efficiency olive to olive by Philaenus spumarius is around 6 to 13%. So what makes Philaenus a very relevant vector of Xylella fastidiosa, for example, from olive to olive, is not the transmission efficiency, it's the vector activity. A very high number of individuals spending on a plant a long time. And what we really don't know is what are the factors influencing the vector activities. So what are, for example, how a Philaenus locate a host plant within the landscape? What are the main cues driving the individual toward the host plant? And once an insect alight on a plant, what are the plant-related factors that permit Philaenus to recognize that as a suitable host plant? So the better the cues, the higher the like vector activity, the higher the chances for the plant to serve as source of the bacterium or recipient of the bacterium. So the characterization of these cues and the cues and the interaction with the vector activity is essential to understand the transmission biology of Xylella fastidiosa, and has also repercussion for the control. So currently, the control of Xylella fastidiosa, at least here in Italy, so is based on the removal of infected plants and on vector control, and maybe mainly on two strategies: ground cover removal and insecticide treatments. But we have two problems. This ground cover removal and insecticide treatments are not a sustainable strategy over the long term. Plus, it seems to be not that efficient at containing the spread of Xylella fastidiosa. So maybe a better strategy would be what we call behavioral manipulation. So interfering with the cues that drive an insect toward a plant or permit the insect to recognize that plant as a suitable host plant to interfere with the plant-vector relation, so with the transmission of the bacterium. So research question of paramount importance is to identify these cues underlying the vector activities. So basically, the main research question that we have here is what makes a plant like a suitable host of Philaenus? What are the cues driving Philaenus toward the plant? And honestly, it seems simple, but it's a very complicated question that requires a very long journey. And we started this very long journey with a much simpler question, just focusing on olive. So for olive, we know that there are olive varieties susceptible to the ST53, the strain causing the olive quick decline syndrome, like, for example, the Xylella Seminar. And two olive varieties have been identified as resistant to the bacterium, which are the Lecchino variety and the FS17 variety, or the Favolosa variety. This looking just at the bacterium-plant interaction. But our question is there also a resistance to the vector? Does Philaenus like, for example, more susceptible varieties compared to bacterium-resistant varieties? And this is very important because of understanding if there is a resistance also to spittlebug would better explain what is, for example, the disease prevalence under field conditions. And to answer to this question, we did multiple trials, choice test and non-choice test. We did also an EPG, electrical penetration graph assisted observation of the probing and feeding behavior of Philaenus on these three varieties, the susceptible Xylella and the bacterium-resistant Lecchino and FS17. And also we tried to explain our data from choice tests and from EPG, analyzing the xylem anatomy of the portion the spittlebugs had access to, so fed on, and also doing a preliminary characterization of the primary metabolites in the xylem sap of plants where Philaenus fed on. So the first test is this choice test. So we started just releasing Philaenus inside a cage with three plants, an Xylella, a Lecchino, and a Favolosa. And we gave Philaenus an observation period of 72 hours. So during 72 hours, we observed where Philaenus was moving. And in this test, Philaenus exhibited a preference, a significant preference, for the bacterium susceptible Xylella. So most of the Philaenus were moving toward the Xylella compared to the bacterium-resistant Favolosa and Lecchino. And the choice was made within the first 24 hours of spittlebug release inside the cage. So in 24 hours, spittlebug decide what are the best plant, they alight on the plant, they settle on the plant, and they remain there without significant movement between varieties. And also we observed a significant preference of Philaenus for the apical portion of olive plants, specifically the main stem, no matter on the variety. So on all the variety, we observe Philaenus on the apical part, mainly on the main stem. And a similar observation was made when we did no choice test. Basically, we forced Philaenus to feed on a plant, whether Oliarola or Lecino or Favorosa, and we observe like the behavior of individual and the within-house preference, which kind of tissue they prefer, which kind of portion they prefer, with an observation period of 38 days. So Philaenus, even in no choice test, preferred the apical portion of the olive plants and mainly the main stem. But another interesting observation comes from the mortality rate of Philaenus. What we observed during these 38 days where Philaenus were forced to feed on a single plant is that there was a higher mortality risk on Lecino plant. There were like more Philaenus dying on Lecino plant compared to both FS17 and particularly Oliarola, where like the mortality risk was like significantly lower. And this observation is consistent with the EPG data. So we use the electrical penetration graph to characterize and analyze the probing and feeding behavior of Philaenus spumarius on the 3 varieties. And what we observed is that there was just one very significant difference between the 3 varieties and the probing and feeding behavioral variable, which is the ingestion of xylem sap. The ingestion of xylem sap displayed by Philaenus was significantly longer on Oliarola, so the bacterium susceptible variety, than both Lecino and Favolosa. So the xylem sap ingestion is basically the yellow part of this cake. The shortest xylem sap ingestion was observed in the Lecino variety. And again, there were no other significant differences. So basically on the 3 varieties, Philaenus took exactly the same time to reach the xylem, to do other kind of activities, and the only difference was the duration of the xylem ingestion. Longer on bacterium susceptible variety, shorter on bacterium resistant varieties. So looking at this data from choice test, no choice test, and EPG, we seek for an explanation using both microscopy to analyze the xylem vessel anatomy and characterizing the primary metabolites present in the xylem sap. First part, like microscopy. We randomly choose like samples like olives and apical portion where Philaenus fed on. We did light microscopy, and then we calculated data related to the vessel diameter, the xylem vessel aggregation, and also the xylem vessel depth. And looking at this data, we observed that the xylem anatomy did not explain the EPG data that we got. Basically, the xylem vessel diameter, for example, was non-statistically different between the Oliarola variety and the bacterium-resistant Lecino variety. So the 2 variety with like opposite xylem ingestion trend. The longest on Oliarola, the shortest on Lecino. The FS17 was instead the variety with like the deepest xylem vessels and also the less aggregated xylem vessel. But these like deeper xylem vessel, less aggregated, seem not to affect the probing and feeding behavior of Philaenus, since we didn't find like significant differences with the other variety and the time needed from the insertion of the stylets to like reaching the xylem vessels and beginning the xylem sap ingestion. Here in the slide, you can see also on the bottom right corner, transmission electron microscopy section that we did on Lecino variety. Here you can see basically what is inside the rectangle on the picture on the left, the trace of the stylets penetrating within the tissues of Lecino plants. And on the bottom right corner, what you can see is like the stylet trace. And you can also observe that when the stylet pass, it seems to compress the parenchymal cell, causing also necrosis of the tissue. So first part, microscopy, it seems that xylem anatomy has no role in the probing and feeding of spumarius. But so interesting data came from the analysis of the primary metabolite of the xylem sap. The main difference between the bacterium susceptible Oliarola and the 2 bacterium resistant varieties, L-Spes and Lecino, were in the organic acid. With organic acid, specifically malic and isocitric acid, being more concentrated in FS17 and Lecino. So the two varieties where Philaenus exhibited a shorter xylem subingestion compared to Oliarola, the variety with the longest xylem subingestion. Another interesting observation is amino acids. So there was no significant difference in the amino acids among the three varieties tested. And for this insect, it seems that amino acids act as feeding promoter. So when the insect sends the amino acid, like the xylem subingestion begins. But then probably from our observation, we think that when the xylem subingestion begins, since like sensing stimuli that trigger the xylem subingestion, then the maintenance over time of the xylem subingestion, so also the vector activity, depends on other metabolites present in the xylem sub. That could be primary metabolites that could be confirmed as organic acids, or could be a combination of primary and secondary metabolites. So this work has been published in Journal Pest Science. Here the reference, and the two main take-home messages are, first of all, yeah, there is a difference in an external difference in olive variety. When Philaenus is given a choice, Philaenus is, in our experiment, going toward the bacterium susceptible. So there is something that Philaenus perceive and that attract Philaenus toward that plant, that Oliarola plant, for example, in our experiment, compare, for example, to Alecino. The second part, once Philaenus alight on the plant and settle on the plant, the decision of whether that is a suitable host or a non-suitable host is dependent on what is inside the xylem sub. So the chemical profile of the xylem sub. Regarding the first, so the external factor driving Philaenus toward a plant rather than the other, there were like several works during the year focusing mainly on olfactory cues, but these work were not conclusive. So there was no association between like a certain particular compound and the orientation of Philaenus toward the plant. But there is another aspect, a fundamental cue, which is important for insects and that permit insect to like locate, for example, a plant, which is vision. And vision and visual cues have never been investigated for spittle-bugs. So from these results, we started basically two projects. The first focusing on vision and the second focusing on, let's call it taste. What's inside the xylem that is feeding repellent or feeding attractant. First part, vision, is a project that is being carried out by Domen Lazar, which is a PhD candidate and a research associate in between the University of Bari and the Gregor Belusic group at the University of Ljubljana. So Domen started by characterizing the anatomy and the physiology of Philaenus eyes. And is like astonishing, at least to me, I'm new to the word of vision. So Philaenus, Domen concluded from his observation that Philaenus has a trichromatic vision. So Domen found three different photoreceptor types with peaks in the UV light, the blue, and the green light. Plus the UV and B sensitive cells have a high polarization sensitivity. This means that, like talking very generally, is that Philaenus is able to perceive colors and shades of colors, and is also able to perceive the polarized light. So probably these data are consistent with our observation on like a preference for Oliarola and Lecino, which is guided by visual cues. Is probably able to differentiate between like different olive varieties because of their spectral characteristic. After that, Domen combined different techniques of microscopy. Here you can see image from micro-CT scan that were kindly provided by Elizabeth Clark from Almeida's group at Berkeley University. So through like a combination of micro-CT and transmission microscopy, looking at the ommatidia, Domen concluded that Philaenus eyes have like a low visual acuity. Basically, Philaenus seems not really able to like perfectly distinguish images with a high resolution, but again, sees colors, see polarized light. So these two cues could be the main driver driving Philaenus toward a suitable host. And currently, Domen is doing like a test using different olive varieties, 16 olive varieties. I'm going to talk about this later, talking about like taste. 16 olive varieties with a different degree of susceptibility to Xylella fastidiosa, and is doing behavioral tests to see whether Philaenus exhibit like a clear choice for certain varieties, and also is characterizing like the spectral property and is doing a polarization analysis to see which kind like of spectral characteristic are the most attractive or the most repellent for Philaenus spumarius. This is the first part, vision. Second part is taste, and there's a separate research project which is being carried out by a PhD candidate at the University of Bari, Zeina Mamouche, and by Maria Assunta Crescenzi, a postdoctoral fellow in between the University of Bari and the Paola Montoro group at the University of Salento. What the two are doing is like expanding on our preliminary finding and extending the observation to these 16 olive varieties with a different degree of susceptibility to Xylella. So according to the literature, this variety exhibit a different population of Xylella and a different symptoms severity. So what they are doing is, again, applying the electrical penetration graph to characterize the probing and feeding behavior of Philaenus on each variety. But this time, the correlation is single individual, single plant, so single xylem sap. So after the EPG from each single plant insect combination, through a Scholander pressure bomb, the xylem sap is extracted, and then non-targeted metabolomic analysis is carried out by liquid chromatography mass spectrometry, like focusing on both primary and secondary metabolites. So the data are extracted and then correlated with the probing and feeding behavior. So the single behavioral pattern and all the behavior of Philaenus associated with xylem sap and xylem sap ingestion. And by now, there are 47 compounds, 47 metabolites that have been characterized. So the preliminary work was done on 4 varieties. Again, Olearola, Lecino, Favolosa, but also Coratina. And the preliminary results that are here of this PCA analysis is that, for example, Lecino and Coratina form 2 separate clusters. And again, as we found like in the first observation on just preliminary metabolites, organic acid as malic acid and citric acid are more concentrated in the Lecino variety that in our first experiment, we found to be the variety that Philaenus preferred the least, the variety with like the shortest xylem ingestion compared to the one that we tested. The olive part is just the first part because we are carrying out also observation on herbaceous host and on the egestion behavior. The egestion behavior here you can see on the right, like EPG waveform, that means egestion and inoculation of bacterial cells. We are carrying out an experiment on herbaceous host using infective individuals. So Philaenus spumarius with Xylella fastidiosa, and followed by extraction of the xylem sap and characterization of the metabolome to see whether also the egestion behavior, so the inoculation of Xylella fastidiosa, is triggered or not by specific metabolites or combination of metabolites in the plant. Obviously, we will keep you posted about all of these activities. And let me first of all thank the amazing Xylella group working in between the University of Bari and the CIAM Bari, and also in collaboration with different institutions, University of Ljubljana, Salento, Potenza, the EPSP, CNR, and the CIAM Bari. And these activities are carried out in the frame of 3 research projects, the project NOSE, COVAXI, and SOS. And in the end, obviously, let me thank you for your attention, and I'm here for questions.
Thank you so much, Daniele. Very interesting talk with very nice results in a very short time, because how many years have you been already in Bari back?
2.5.
Yeah, so you have been quite busy. I see. So remember